Welcome everyone into this video tutorial of COVID-CDR. In this video, I'm going to show you how to quickly use COVID-CDR for exploring a drug combination of COVID-19 drugs and their network-based mechanism of action. There are a couple of tabs available in this particular page where we have a search page where you can select two drugs and search their combination. You can observe their pairwise drug similarities. You can choose their any particular drugs and the corresponding drugs that are highly similar, and you can choose their couple of options based on what you want to select their significantly similar drugs. You can see some of the curated combinations that are collected from literature or third-party resources. The drug pairs that are in clinical trials, the drug pairs that are FDA approved, or combinations that are FDA approved, but for the different indications. And you can also see some of the drug combinations that are conducted on different specific cell lines. Obviously the drugs that are involved in this particular project. You can also download the data that we have either generated into this project, or we are we're showing into this interface, or we have generated as an intermediate data. You can all collect them by downloading this through this link, and you can download them in various formats. So let's have a quick look how we can explore COVID-19 drug combination using COVID-CDR. Firstly, we have two drop-down menu, which will allow us to select two drugs from like drugs of our interest. So importantly, the list of drugs that we have considered initially is limited to 867. So in the drugs that we would like to demonstrate here, they are listed with their generic name followed by their classification. So what you need to do is just to click backspace, so the text would be removed. Now, if you want to type your own drug of interest, for example, remdesivir, you start typing rem, and then the search will be filtered accordingly. So here I can see the remdesivir is coming when I have just started typing RMD. So REMD. So if I click this one, then this drug would be selected, and similar option would be available for the other drop-down menu. Now we will be clicking search to construct the network-based mechanism of action network. So here this network is now getting generated, where we have nodes and we have ages. So nodes are basically different genes or proteins from different organisms, or the drugs that we have interested. Here, red color genes are basically they are from SARS-CoV-2 organism. Black color genes are from SARS-CoV organism. Blue color or green color nodes, they are human genes, and the pink color nodes are the drug that we have initially started with. So basically, this network that we are seeing, it is an induced subnetwork of a super network, which is composed of many other networks, like host pathogen networks between SARS-CoV-2 and human, SARS-CoV and human PPI network, and drug target network. So here, this network is highly interactive, which means if you mouse over on any node, you can see some information, like if that's a protein, then you can see their name. If that is their drug, then you can see the name, class indications, and some of the proximity measurement that we have derived for this particular project. And they are the proximity scores, either functional proximity or topological proximity in the context of COVID-19 drug modules. Not only the nodes, but the edges are also interactive, which means if you click any of the edges, then we can see some sort of this edge is queryable. So if you click them, then the PubMed would be queried with these two incident protein names. or any of the names, like it could be drug and protein as well, but which literature talks about them. So if you can see in this manner, it is querying PubMed, and the result is retrieved. So you can click click this particular literature, and it will redirect you to that page. So now in this particular GUI, we have a couple of features that we have shown here. Firstly, the database icon. If you click that one, then you can see the same curated combinations that we have seen in the first page. The combinations that are available in clinical trials, and the combinations approved by FDA and all that. So the information in the first page that is still available here. And these particular combinations are quite actually interactive. So if you click the row here, then the corresponding combination will be selected, and their network will be visualized accordingly. And same goes with the other tables as well. So so this is what what is generated for remdesivir and interferon beta 1A. Now, if you're interested into a new pair of drugs that you wanted to make a query with, so you can select this particular button, where you can select two of your drug of your interest. And then if you want to add some new drugs on top of what we are seeing here, you can click plus, and you select the drug of your interest and click plus button here. So that means the network would be regenerated because it's adding a new drug, and the dynamics would change, and all the different metrics would be changed as well. But actually, that is making the network a bit more complex. But still, I mean, the high usability of this interface is that it can, although it looks like a bit complex, but it shows us like some of the crucial quantification. For example, the proximity scores, network-based separation score, which we can use for your further research. So, for example, here in this panel, we can see their pairwise drug similarities and their network separation scores. So network separation scores, all these values, basically they are between 0 and 1. So higher values means like, for example, if the higher score in network separation, that means two drugs here, they are basically further apart into this network. So they're highly separated. And the chemical similarity indicates how these two drug structures are similar, and target sequence similarity means how these two drug target sequences are similar based on the sequence similarity, and pathway similarity and GOTAM similarity. Basically, they are the similarity measurement in the context of their functional enrichments. So this panel is very useful. Now, if we can also derive some sort of we can also show some sort of additional metadata for either proteins or the drugs. So if you click any of the nodes here, if that's a protein node, then the panel, the information panel would be pop right, and then it would show us a list of enriched Go terms for incident with this particular protein that we have selected, and also it will also show us what are the incident proteins or nodes it is connected with, and the corresponding species information. If we click any particular drug, so in this case, let's pick dexamethasone, then we can see a whole lot of information for this particular drug. First of all, the drug structure that would be generated from mol view. You can pop out this particular view to see it in a larger view, and you can also see the periodic table in order to help you to understand the different molecule color of different molecules in this particular view of the drug structure. You can also see the proximity measurements that is generated for these two drugs. You can see some of the side effects available for these drugs from the CIDR database. Their adverse drug interaction, some physical, like some of their metadata, like physicochemical properties, pharmacological properties, and all that. Now, so the view that we are seeing here is the combination, like the combination of a list of drugs and their corresponding network-based mechanism of action. Now, initially we have started with a fixed set of drugs, which is 869, but we have added a new feature to go beyond that, which means we can add new drugs if we like to add new information on top of it. So if you want to do that, you just click this customize button, where you have an option to upload like a file from your own, which we have shown an example. All you need is just to click this link and the file will be downloaded, and you can browse that file and click process. So the file will be processed, and the new drug and drug target file will be augmented with the existing visualization. So what this file format actually does, I'll quickly show you its format. It's a new drug target file. So this is how this file would look like, where we have a gene name, UniProt ID, species, drug ID, and drug names. So this is the format this particular interface actually requires at this stage. So you have drug targets here, the drug target gene names, and the UniProt ID, which species it belongs to, and the drug ID that we are interested here, and the corresponding drug name. So in this case, EGFR has been targeted by 3 drugs and their corresponding drug names, DR1, DR2, DR3, and ERBB2. ERBB2 genes also the same. So here, this is the format we would require at this stage. So if you follow this format and you just upload that, browse that file, I will do that from here. Drug target file. So I'll upload that and click process. So once I click process, so as I said before, the new drugs would be added on top of what we have seen here. Just click process. So since this is going to be an expensive operation, so it's showing us a bit of some sort of message that whether you want to still proceed with this or not. If you don't want to proceed, you can click cancel anytime. But if you click OK, this is going to take a while. So here, while this is computing, so here once we start processing, the new option has been generated dynamically, which means if you want to restore the defaulted or not, which we're gonna demonstrate very quickly. Let's first see the new generated network, which is currently being built at this stage. Yeah, so it has been generated now. So as you can see, this network has become more complex to follow. So here, these are the 3 new drugs that we have added: DR1, DR2, and DR3 on top of what we have seen before. So obviously, because of addition of the new 3 drugs, the network would, like the interface would respond accordingly, which means you have the new drugs added into this panel, but the network separation has not been generated because they are generated on the fly. They don't have any network separation scores calculated, or their similarity scores as well, because these data are pre-computed as they are not generated on the fly. So they will not, at this setting, we will not see such information for newly added drugs, but we will only can see how they look like into the context of the new drugs. So if you really want to go back to your previous network just before adding this new information, you can click this checkbox. It will destroy the previous view, the current view, and restore the previous view. So yeah, so the previous network has been restored. So that's all for this video description. I hope this video description is useful. Thanks for watching.